Okay. So now for this second talk, we have Lucia Melloni that has her lab at the Department of Neuroscience, Max Planck Institute for Empirical Aesthetics, and is also affiliated to the Department of Neurology of NYU Grossman School of Medicine. So Lucia, thank you very much for being here, and the audience is yours. And we don't hear you right now, Lucia. You're muted.
Can you hear me now?
Now, yes.
Okay, so sorry, I was just saying that, thank you for the invitation to you and to David, and that it's a pleasure to be after Tristan, because I want to try to somehow build on the things that, he presented before, but make them, instead of a sequences of beep, beep, boop, sequences of syllables and hopefully language. But in essence, it's the same thing, it's the sequences of something. And the talk is trying to actually go, as the title says, from continuous stream to segmented units, and understanding like how we structure events both for cognition or perception, if you want, and memory. And the problem that I'm trying to get at is, is something such as this, which is, how come that we can understand that this gentleman is dancing somehow? And what I'm going to be showing is data and beautiful papers published already by these 4 people, Naiding, Radhika, Simon, and Valerie. And you're going to see their pictures as I go along in the talk. So I want to start by bringing us back to what I think that is a problem behind predictive processing, and in some sense, statistical learning, which is the fact that sequences are ubiquitous. We live our life, like in something such as music, where there's of course sequences and structure. The same thing actually happens, for language. And one can actually argue whether or not the same syntactic process is similarly available for music and for language, but at least minimally one can say that they share the same. They share the fact that there's structure there. The same thing perhaps also happens when we are having motor sequences, right? And the latter one is about spatial navigation. So you can also think that, how can I get from place A to place B? I actually need to follow a sequences of path. I need to remember the order in which those happens. And if I don't remember the order, then it will actually end up somewhere else. So in essence, what the way in which we're thinking about this problem is perhaps there is a basic computation that is underlying all of these different seemingly, disparate cognitive phenomena. And it starts with, like the simple realization of the fact that once upon a time we were all a baby, and life was continuous and also mumbo jumbo. We didn't understand anything. So the first thing that we actually have to solve was to go from these continuous waveforms, as for instance in this example from Patricia Kuhl, there are no silences between the words. That immediately tells you that even though in your head and in your mind, you're putting together these silences and you think that I actually speak, with chopped words, and with silences between what I'm saying, if you look at the waveform, you will see that they either not there, or if they are there, they are in places where you actually don't perceive as there is a pause. So the first problem the first order problem is like, how do we go from this continuous stream into units for the mind that we can consume and then we can communicate? And what I want to bring about is the fact that this happens both for perception and for memory. And once we discover those units, then of course there's something simpler that we need to solve, which is the fact that within a unit we need to integrate, and across units we have to segregate. So immediately brings about the problem, like how do we actually put things together and how do we separate? Then the next, which I think is already touched by Tristan, is the fact that perception, language, and a lot of these things are actually hierarchically organized. Take the example of language. We go, like from tones to from syllables to words, like to phrases to sentences and so on and so forth, right? And they all actually have an embedded hierarchical structure. We understand them all together. So there's something that needs to be explained also in terms of the windows of integration that we have for these very small units and the very large units. And finally, because that we actually live in a world in which probably sequences are important, we need to organize time. Because it's not the same if I start with the end of the sentence or if I start with the start of the sentence, right? Or the same thing, think of spatial navigation. If I actually missed up the order of the things, I end up somewhere else, right? So these are the 4 topics that I hope to cover in the next, say, half hour or so. So let's start with the first one. As I told you, look, once upon a time we were all naive and we didn't understand anything. The world sounded something like a speech waveform that was completely continuous. But eventually, we actually we learned language and we started segmenting it in syllables that would make sense to us. Then, when we segmented those in syllables that they make sense to us, we also started building up words. Actually, we started compounding from these, very elementary units, actually high order units, as in this case, the words. And then, when we were whatever to it, when you can discuss at what age during the infancy that happens, but we started building high order units, such in this case phrases, and then sentences and paragraphs and so on and so forth, right? And this all happens really simple. You don't have to, pay attention to this. This is one of the things that, it's interesting because it just happens to you automatically, as we would say, right? It doesn't seem costly, right? Yet the question, back in the day was, how do we actually put together these high order units? And we started from the CNP that, Tristan was actually discussing before, which is the idea that there might be temporal windows of integrations that will be tracking each of these units. And the, a very naive idea at the time, with Nye and David Popple at the time at NYU, was that the length of those windows would actually encompass the length of the units that is being integrated. So very short for syllables, longer for words, even longer for phrases, and yet longer, for sentences. And here you can see why it's so nice to actually have a 1/f distribution, because this means that your brain can actually be in tune with all of those different windows of integration, right? And to get at this, what Nye did was to create completely artificial languages. So in this case, I don't think that actually I was presenting sound, so it doesn't matter because it's in Chinese. So what Nye did was to create in MacTalk, completely artificial syllables, and in this case they were in Chinese. And what was the trick was that each of those syllables would last 250 milliseconds, right? They would actually be a word in Chinese, and if you put two together, they will actually be compound a noun phrase in this case. Let me see what you can see in my laser. So if you put two syllables together, this will make a noun phrase. If you put another two syllables together, it would be a verb phrase. And if you would put a noun and a verb phrase together, it would be a sentence. And here you can see why it's so important, the fact that the syllables were 250 milliseconds. Why? Because then you would have 4 in a second, so 4 hertz, 2 in a second, so 2 hertz, and then 1 in a second, so 1 hertz, right? So now you can tag whether or not there is a resonant frequency in the brain when this happens. And what I did in this case was just to present this. These were like 12 sentences in a row. The subjects in this case would be in the MEG and would just listen to this. They would just do nothing, right? And the question was, can we actually find peaks in the MEG spectra at each of these integration units, so to speak? And sure enough, we found it, and that was super. I thought it was never going to work, to be honest with you. But Nye was definitely determined to actually, try it, and he found it. And the first time we saw this result, I was like, oh my god, this is just really weird. I didn't think that it was going to work, but it did. And as you see here, you have a very strong pick to the syllables, then to the phrases, and then to the sentences, and then you would say, well, this is trivial. What if there is just energy in the stimuli? So what you're just tracking is literally energy at each of those units. Well, that's not the case, because you can turn, any one of these subjects into a naive subject. So now you present these Chinese materials to you, hopefully English speakers or Spanish speakers such as me. You understand nothing. You get the peak at the syllabus because we know there's energy at this level. But if you cannot integrate in your head at this at the phrasal level or at the sentential level, now there is nothing that we can actually recover, right? So this is not merely the fact that there is no energy here, the energy that is put because your head puts together a unit. Now this is actually interesting in light of what Tristan was showing. The question, of course, we present completely artificial materials, but if the hypothesis is correct, we should actually have these tracking windows depending on the length of the stimulus materials, or depending on the length for which the subjects are integrating. And this is actually the case. So now if you present it in the time domain, if you have a sentence of 4 elements, then the peak is here, 5 elements here, 6, 7, and 8. So as you see, you have a nice range in which you can actually track the different lengths. Now, this also, I guess, an interesting light of what Tristan was showing. This is, like if you now looked at the time at which a unit is, stops to be integrated, and you look what happens before, you have a nice decrease in power and then an increase at the time. So you can actually, here we can discuss later on what part of this is a CNV versus a, prediction error, because probably both of them are actually at play here. There's something even nicer, because if this hypothesis is correct, there should be integration across integration across units. So now think about a sentence that actually has 7 words, but can either be compound of a 3 syllables noun phrase and then a 4 syllables verb phrase, or a 3 syllable noun phrase and a 4 syllable noun phrase and a 3 syllable verb phrase. So what you have here, first of all, is this, like arc, which represents the fact that it's integrated over the 7 words. But at the same time, you have different peaks that reflect here I'm integrating over 4, here I'm integrating over 3. So you can actually see both of those working at the same time, right? All right. What is probably even more interesting about this is what happened when we looked at, so these are all imagery recordings. We don't know actually where any of these signals are coming from. So we repeated the experiment with epilepsy patients. These are patients that have electrodes in their brain. So it's a window in which we can, this offers a window for us to look into the brain with very good spatial and temporal resolution, but it's only in the case of patients, right? So each of those dots represents an implantation scheme for one patient. The color is, each patient is color coded, and as you can see, we have a very nice coverage about from the STG and the infrafrontal gyrus as well, which are areas implied in language, presumably. So what did we find? We find electrodes that had different temporal tunings. So one of those had a nice response to the syllables and a nice response to the sentences, but almost virtually no response to the phrases. Yet there were some other set of electrodes that would have responses to the syllables and to the phrases, but not to the sentences. And the third, which is probably even the nicest and more interesting one, is it would have no responses whatsoever to the syllables, but only to the higher order units. So in this case, the phrases and the sentences. Where are they localized? In, interestingly enough, at least for this experiment, I'm going to show you later that it's not the case for statistical learning, and they are actually in similar areas, right? So as you can see, they are all posterior STG, infrafrontal gyrus, and actually they happen to be more or less intermingled, right? But they encode different information even though they are in the same area, right? Another interesting aspect of this technique is how reliable it is in the single subject level. So these are the single subject participants, both for the native language as well as for the unknown language, and each of these lines represent one participant. And as you can see, in all of the ones that are actually native speakers, we have a nice response to the sentences and to the phrases and to the syllables. And if they don't speak the language, there's just, simply no response. Because this is so reliable on the single subject level, now we can actually take it the next step, which is, can we try to use this technique to evaluate whether or not somebody who is unresponsive and conscious, presumably because it doesn't have any overt motor response, is or not comprehending language? And this is something that Radhika did with Damien Cruz in Birmingham. What she did was to evaluate, in this case, 28 patients that were 2 to 7 days after sedation, but they were completely motorically unresponsive. So for all that, would you think overtly, they would be unconscious. And she just put, like an EEG cup, as you can see here, presented these sentences, with, that would, you would expect the response to the word, the phrases, and to the sentences, and look at the EEG response. And the hypothesis that we had was that presumably those patients that, would be conscious would have a response to the phrases and to the sentences, because they would be understanding the language even though they would not be able to overtly react to us, right? So this is what Radhika did 2 to 7 days after sedation was removed, and then she contacted the family 6 months later, right, and asked for the GO scale. So just for those of you who don't understand, like you can think of this as a conscious meter, from being pretty much unresponsive to being pretty much really well, very well responsive. And what she could do is that she could predict based literally on the peak responses of these two responses, in this case to the phrases and to the sentences, whether or not the patients later on would actually recover, right? And this was above and beyond what could be predicted based on the clinical evaluation as well as on the CT. So it offers a simple 20-minute EEG showing passively, actually offers the doctors in this case a possibility to evaluate whether or not patients are responsive. So I think this is, from a different point of view, it's an interesting it's an interesting way to think about these techniques. All right, but okay, so from this, what I want to bring up as an interim conclusion is that we know that this is a very robust technique. We have, we ourselves have tried it with Chinese materials, with English materials, or have extended to Hebrew, to German. It works not only in our lab, in other labs, which is really good because it replicates. We can show that it really tracks the hierarchical level of structure, and it's not confounded with simple acoustic responses. And we can use this to hopefully as a prognostic tool in the case of unresponsive patients. So I think this is, at least to me, like this is the probably most exciting result. All right, but as I started, what is nice about this experiment, it shows us that if you have learned the language, we can track that your brain integrates that. But once upon a time, we were all naive, right? And the question is, can we use this frequency tagging to track how subjects are learning a language, right? And here's where statistical learning actually comes about. As you know, the major challenge is, is to evaluate how babies are finding these units. And one, for those of you who are familiar with statistical learning, this is the seminal paper by Yamil, where she had this brilliant idea of asking, like, can babies just simply be like fantastic bootstrappers? They would actually compute the statistics in their language. So if in this case, for instance, to was always followed by ki was always followed by bu, but bu was followed by either gi, go, and ti, then the babies would just in their head compute the statistics, put a scissor in their head, and then say like, oh, here the word stops, and here the new one starts, right? And the way in which she did it is, to create 4 artificial words, the words compounded of 3 different syllables, right? This would be one word, this would be another word, another word, another word. And she presented 8-month-old babies with these materials for 2 minutes. And she was able to demonstrate that very simple exposure, in this case, of 2 minutes, already create or already presented behavioral signs for babies that they could actually understand and extract these statistics. So that we know that actually happens. It was, this is a 30 years of really, really flourishing research showing that babies can not only extract, in this case, words, but even more excitingly, that this seems to actually be a basic computation for the brain in which the same machinery can be deployed to understanding events, right? And this is actually a beautiful work by Nick Brown and others, where, for instance, you can now pair 2 different fractals, and if you pair them statistically, then babies, patients, and like adults learn that these 2 make a unit, these 2 make a unit, and so on and so forth. So what is relevant about this is that this seems to be a basic computation that can be deployed to understand words as well as events in the larger sense. So this was. It's a really nice area of research, and there's you can do this with natural images and so on and so forth. Yet there's what I think that simple basic questions that still are untouched, which is one of those is, what are the neural circuits responsible for the discovery of those events? And this has something to do with the fact that you want to know, like, how are subjects learning as you expose to the materials, not just a before and after. Which is what has been typically done before. And also, if there's a basic computation that underlies learning of the structure at the visual modality, in the auditory modality, or in other modalities in which this can be at hand. And finally, what information is actually represented in those events? And I will show you later on something which I think is really important, because in these very simple and minimal sequences, actually you would be surprised how complex they may end up being in terms of the amount of information that can be extracted. So what Simon did in this case was he took the same materials as in the Jenny Safran, so in this case 12 syllables. And the idea here was, if we present these materials, there will be a resonant frequency at the level of each of these syllables. And this would actually occur in the random stream, so where there is no structure whatsoever. But if we present the words in the statistical learning form, so to speak, so 2 is always followed by P is always followed by rho, and so on, eventually over time, subjects would learn this higher order structure, and we would actually frequency tag them. So there will be a response that actually is lower, 3 times lower, because the subjects are integrating over 3 units, right? And so now, if this is true, what you would do is that you would actually say, well, there should be a response at the frequency, which is at 4 hertz, and response at the word, which is 1.33. Again, we took advantage of patients that have epilepsy in this case. These are, as I told you, patients that actually are waiting in the unit for seizures to come. We exposed them to these materials. They actually had to do, the only task was to press a button every time, and a very rare repetition of a syllable would occur, but we didn't tell them that there was a structure whatsoever, nor did we ask them to pay attention to the structure. This is completely passive, completely implicit. And these are 16 patients that we evaluated, again, color-coded the different patients with the different electrode implantations. And nicely, what we observe is that in the random stream, there's only a response to the syllable, but in the structure stream, we have now a very nice response to the words, right? And this is, again, 2 minutes of exposure times 5. So this happened in 10 minutes, just for you to see like how quick this actually happened. Do we observe something similar for the visual modality? Sure enough. So now what we did, and I can explain later on why, instead of actually having triplets, we presented only duplets. So in this case, are pairs of fractals. And you would expect, again, a neural response for each of the fractals if they are random, but also response that is slower, half the speed, so to speak, if subjects are learning the pair structure. And again, we repeat this in the ECOG. These are the electrode implantations. And sure enough, now you can see this very nice response to the random stream, in which you only have response to the fractals, but nothing to the pairs. And in the case of the structure, you have, again, a very nice response to the pair as well as to the fractal. And again, this is 10 minutes of recordings, just for you to see like how quick this can actually happen. I remember that I had shown you before that in the case of the language, we found these 2 kind of like temporal windows of responses, one to higher order units and also to lower order units. Well, interestingly enough, we recovered this. This is not something that is we predicted whatsoever. This just, we just found this. I just want to be clear about this. What we observed was 2 kind of electrode responses. So one set of electrodes that would not respond to the syllables, but would have a nice response to the word. This we call word only. Another set of electrodes that would have a response to the words as well as to the syllables. And here you see that actually they do not co-localize. So the electrodes that respond to the syllables, or in the input and, so to speak, like the high order unit, localize around STG, whereas these other high order ones, interestingly enough, actually are outside the STG, right? And even more, interestingly, we actually found that the same temporal tuning for the visual modality. Now, again, in the case we found electrodes that would respond only to the pairs, some to the pairs and to the fractals. Now, as you would expect, since these are visual stimuli and not auditory stimuli, the pair and image actually localize in the visual cortex, not in the auditory cortex as you would expect for this other one. But again, the same patterns occur in terms of them not being in the same areas, right? So the high order ones, so to speak, are in different areas as the ones that respond to the initial units. All right. So with that at hand, we actually then ask the next question. So it's true that this is a very simple sequence. It's just 3 different, like syllables, times 4 words. Now, what could the subjects be learning? And what I want to bring to your attention, guys, is that this, frequency tagging response that integrates over the whole unit is equally compatible with 3 sort of, for information that the subjects might be learning. One of those is transitions and timing, which is I think what Tristan was showing you before, right? So you can think that the first syllable is unpredictable. The second one and the third one is actually predictable. So you can just the subjects can literally be computing like prediction, prediction, prediction error, prediction, prediction, prediction error, prediction, prediction, prediction error. And that will actually yield the response at the higher order units. They could be learning something actually far more complicated and far more interesting. They could be learning the position, the ordinal position. meaning in this case that they would put together the first, so in this case, tu, bi, go, and pa, and they would also be coding for the second position, so pi, da, la, and do, and for the third one, ro, ku, bu, and ti, right? So these are really abstract coding of literally order, first position, second position, third position, regardless of actually what the word was, right? And the third is the chunk. They could actually be learning that it's important to have an order, but like a word is a word. If I say house, and if I then mix it with something else, it's not the same word. So you actually have to know the order as well as the syllables. So in this case, I would be learning the chunks themselves, right? And any of these three would equally reflect the frequency response that we had in the electrodes before. So to get at this, what we did was representational similarity. So now you take the responses to each of those syllables and you ask, is there any ordering? Is there any do the syllables cluster in a particular way that reflects the coding, right? So now we looked at the electrodes that would respond to the syllables and to the words. These are the two different dimensions. And what you can see here is that they very nicely have a distinction between the first syllables and all of the others. So they're nicely coding for transitions and timing, right? If you want, statistical learning, so to speak, in that sense. Now, the other electrodes, in this case, the ones that were responding to only the words, right, have in one dimension, they respond to the order. So in this case, first syllable, third syllable, second syllable. And in the other dimension, they nicely code for the words themselves. First word, second word, third word, fourth word, right? And these electrodes do not code for the transitional probabilities. I just want to, we tested that. So these are independent coding, so to speak. What happened in the visual modality? We actually find, of course, the electrodes localized in a different place. So in this case, now we have here the electrodes that are responsive to the pairs and to the images. But again, and even though they localize in different areas, they seem to code for the same as the ones in the auditory, which in this case, transitional probabilities and nothing else. Now, the electrodes that would code exclusively for the pairs and not for the fractal have again the same type of coding. So in the one dimension, they would code for position. So in this case, it's first position and second position. And the second dimension, they would code for the chunks themselves. First word, second word, third word, fourth word, right? Now, what happens in the hippocampus? The hippocampus cannot care less about, in this case, statistical learning, so to speak, so transition probabilities or order. It only cares for what it saw, meaning that it cares about the chunk, right? So this is one word, another word, a third word, and a fourth word for the auditory and for the visual. So the hippocampus only represents, well, at least in our hands, it only cares about what is the specific chunk, right? And what I want to conclude with this is to, hopefully I was able to, give you in a quick time, like the evidence that I think that this frequency tagging is actually pretty robust and could be a very useful tool to track not only the outcome of the learning, but also the process of the learning. But also that once we tag those electrodes that actually are that have learned, we find multiple computational systems that encode information at the same time at multiple levels, right? And I think this is an interesting result because there's a lot of disputes in the statistical learning literature whether subjects learn chunks or order or what have you. I think that it seems, at least from this result, that the brain doesn't have to choose among those alternatives. It just learns everything, and why not? Right? And something that I want to just bring to your attention, the fact that these are patients that have epilepsy, so they have very bad episodic memory, and nonetheless they all showed very good frequency tagging, right? So we can discuss later on I think Nick Brown, a good friend and a collaborator in this paper, has a very nice, and Anna Shapiro as well, a very nice hypothesis as to this a complementary learning theory also within the hippocampus with two different pathways, coding for statistical learning versus episodic memory. So, so I hope that I convince you that you can use this frequency tagging to tag words and events in the larger sense, and that this is true, that we could use these kind of techniques to then ask questions about, what happens in babies, what happens in other animals, what happens in unresponsive patients. So I think that it opens an exciting window to do, to allow for interesting interspecies comparison. And if in the next, can I have 5 more minutes, Yamil, or not? Because otherwise I can stop here.
Sure. No, I mean, you can go on. We can chunk out a couple of minutes from the break, which is what comes next. So go ahead.
Okay. So what I want to now switch gears and ask about what happens. So all that I show you now is what happens, hopefully, or seemingly on perception, right? But the question is, what happens in memory, right? Because also the way in which we perceive things may actually have a consequence for the way in which we remember things, right? And a very classic example is this idea of spatial navigation. So your memories for objects within a room is far better than for objects that actually occur across rooms, right? So what this tells us is that the mere fact that we have an event, in this case like a spatial event or a spatial boundary, creates or chunk or creates separations in the memories that we have. And so if this is true, then you can also ask the question, can any event boundary, not just spatial event boundaries, this is the so-called doorway effect, by the way, any event boundary have alteration on temporal order memory? And this is the work that Valerie Poo is doing in the lab. So what Valerie did, and this is inspiring from a beautiful work from Leila Davachi, was presenting 12 unique images to subjects, 36 of them in a row, and they would actually have a color in the background. And this color in the background would create a boundary, so to speak, a perceptual boundary for the subjects, right? They would actually have, 6 of these images would be color coded in one color, so say in this case red and then blue. And she would also have the same different images, but the same, so to speak, like 36 sequences, where there would be no boundary in this case, so the same color, right? And she asked a question then to the subjects after the encoding phase, can you please tell us which of these 2 items comes last? What is the last one? And by doing that, she's asking for the temporal order. And the question that she asked was, is the mere fact that I have a boundary in my perception at encoding, will that affect the way in which I organize my memories later on, right? And so she picked, of course, specific events. So in this case, for instance, 2 and 6 that are within an event, or 5 and 3 that are across an event. So in this case, the across an event is literally just a change in color. But for the subject, this is completely, unrelated. And then she, of course, took the very same serial positions for the condition where there is no boundary. And as a nice control, what we observe is that if you have like the same serial positions for, say, like within an event versus across an event, you have no differences in terms of accuracy, in terms of recognizing temporal order. But now what happens is that the mere introduction of a boundary creates an improvement in the memories or in the temporal order memories for items that are within a boundary, and actually a decrement when you actually cross a boundary. So the mere fact that you have that you create the chunk in your memories helps you in recognizing the order for things that are within that event, but also screws you, so to speak, for recognizing the order for things that actually are across a boundary, right? And this is a real effect, and not just the so-called like temporal distance effect, which is another effect in memory, which it's very simple to understand, which is the fact that the more time passes, like you can think of memories as being the memories can be interfered just because something is close to another, right? So the more time passes between one event and another, then the easier it is to recognize it, just because we can separate them better. So what then Valerie asked was, is this boundary effect different from this just mere temporal distance? And the way in which you can get at that question is, again, presenting the same 36 sequences. And now in this case, she only presents 4, and she color codes only 4 of those. And she picks specific, pairs. Either this one, which represent within a boundary, which 1 intervening item in between, 3 and 1, which represent across a boundary with 1 intervening item, or across a boundary with 3 intervening items, right? And then she asks the same question. So she replicates the results as before. Temporal memories for within an event with lag of 1, so with a lot of interference, is much better than for across an event. But it's still being much better when you actually have a lot of intervening items. So what this tells you is that, okay, the memories get recovered, as compared to 1, but they're still worse, as in the case when you actually are within an event, right? And in the interest of time, I will skip this, but she has a nice computational model that we can, talk about that later on, that explains that in terms of, like a temporal ordering signal and a reset. It's a reinstatement of the context signal at the beginning of each of these events. So in synthesis, what this tells us is that the mere fact that we chunk our perception also has a consequence for how we encode those memories. Right? So these chunks are not just nimious, like they do have a perception, they do have consequences later on. And with that, I hope that I show you, evidence for how, we think, or not how, but just some electrophysiological evidence for, how we go from continuous signal to, segmented units, both for perception and for memory, that there is integration segregation across those, and that, we can track the hierarchical organization as well as the temporal order. And with that, I want to thank the people that helped me, with all of this, Valerie and Simon, and of course, David, Nick, Damien, Sharon, and Oren from NYU, and you guys for your attention. And I'm excited to hear your questions.
Thank you very much, Lucia. So, questions from the audience or panelists? Okay, we have a question by Claire Press. You can ask out loud if you want.
Hello, can you hear me?
Yes.
Yes.
What I was interested in, so the evidence you were showing for these frequency peaks at 124 Hertz, that's all about relative frequencies. Obviously, we can listen to videos at double speed and they're fully comprehensible. What I was wondering was, what does the generalization decrement look like? So, I mean, if you were to just play your videos, your auditory streams at double speed, do the peaks just automatically shift to 248? Or is there generalization decrement, and where does that come?
Let me show you, because that's why I probably, I gloss over this too quickly, but that's why we did this experiment. Whoops. Can you see my screen?
Yeah.
So in this case, so of course we didn't have it monochronous, but we actually now have a sentences that will either have 4, words, or 5, or 6, or 7, or 8. And if this is true, then you would expect exactly that the integration is up until the point at which, you're still integrating, right? So of course it's not linked to a specific frequency, but it seems to be flexible depending on actually how you integrate.
Yeah, okay. Presumably we do learn, though we have a contextual cueing, that syllables typically come in at 3 Hertz-ish. So is there no sign then of being better at those frequencies?
For what? For the higher order units?
For syllables.
Well, so we actually have pretty good tracking of syllables regardless, right? So, this is what we have observed all the time, but also there is energy at the syllable frequency, right? So, so we can track the syllables no matter what, right? The more interesting bit was, can we track, phrases and sentences for which there is nothing in the input?
Yes, yeah. Yeah, no, I understood the main point. It was more that I assume it does come into contact with the learning environment, with the rates that that usually comes in at. Correct.
So I fair point. I think the very more interesting question is, like, what happens if you really slow down the input more than if you make it faster, right? If you really slow down, can you not integrate? We haven't tried that. I think it's an interesting question for sure.
Thanks.
Great, thank you, Claire. Now we have a question by Maria Chait. Please go ahead.
Yeah, thank you, Lucia. That was a really excellent talk. There might be my question is about a detail that you might have mentioned and I missed, and that is in the ECOG study, when you compare auditory and visual patterns, why did you choose, so you have 3-syllable patterns for hearing, but only 2-syllable patterns for vision? Is that because you played around and vision was more difficult to?
No. I have, I can show you later on. Let me, perhaps you can. So no, it was, because we were, so since these are patients, and sometimes we wanted to make sure that it works for the patients. So, the fact was that, if you present, like 3, it would actually be probably too fast, is what we thought. So we were scared. So we only presented 2, but kept the sequences, like the same, right? Just because, like you get only one, shot with the ECOG patients. But then we did this in the EEG. And so here I'm presenting you, these are pairs and these are triplets. So, you get it no matter what. This is that we were scared with the ECOG. So it's not bounded to the, yeah. But it may still have a limit, right? So we only have tried duplets and triplets, right? It may still have a limit.
Oh, thank you.
Great, thank you. Now let me pick one question from the Q&A from Wine 2. In terms of learning, I'm very curious about how entrainment evolves over the 10 minutes block. Is there any single trial analysis that allow us to ask questions such as whether there's a critical point?
I mean, so perhaps, except that, like, so what happens, and this is the message. So we, the reviewers press us on that point, and we didn't want to give in because I think that there is limitations of the ECOG, and I will explain you those. So in the ECOG case, you have serendipitous recordings, so they just happen to be wherever. You cannot really say like I want to have an electrode here, right? So then, and each electrode may actually learn at a different rate, right? It's not that simple to do single trial analysis or single subject analysis in the ECOG. What we did nonetheless was to say, like, what happens if we look at all of the electrodes that we have and all of the patients that we have? And what you can see here, this is the number of pairs for the random sequences. So that there is, there is no learning whatsoever. But, and here shows you like what is the progression? How many pairs would you need in the case of learning to be statistically significant? Right. So we know that here in this case we need something like 400 pairs, but this of course is the higher estimate. Now here you can actually look at the same data from a different point of view. So how many of those electrode pairs showed responses? For instance, when you present like 100 pairs or 200 and so on and so forth. So most of them actually learn pretty quickly, which is what you show here what is shown here in this histogram. Right. Then of course there's a tail. But it's hard to know where it's a tail based on, like electrodes more in frontal, so because presumably what, one would expect is that, perhaps, auditory cortex like SCG learns faster and then later on IFG or the other way around, God knows, but, that the learning rates may differ in the cortex. Right? And this is the latency, by the way, so you can, this is latency color coded, right? So most of them actually are pretty quick, as you can see.
Good, thank you. So let's have one last question before the break from Luisa Bogarts. Luisa, do you want to ask?
Yes, you hear me?
Yes.
Yeah, Luisa.
Cool. Really nice talk, Lucia. Thank you. I was curious, so you showed that the hippocampus is really like a chunker, or it's representing the chunks, but then also both in the Ding paper for language and then also in the Henin paper, there seem to be neurons in cortex that are then specific to the word level, right? Or the pair level in the visual modality. So I was curious if you have any data on patients that have both modalities of, say, neurons that are representing a chunk independent of modality, or does that seem, is it specific? Like, do you have chunk neurons that don't care about what the content is?
Let me see what I understood your question. So are you asking whether there is like a supramodal integrator? Something like that, so that would integrate regardless of whether I'm presenting something in visual or in the auditory modality.
Yes.
Yeah. Okay. So we do have evidence for that in the ECOG. So okay, let me back up. The problem that we have with this data is that we have only I think like 6 patients in which we passed the 2 experiments.
Mm-hmm.
So this comes from different populations. So we actually found some electrodes that would actually code for both, but we cannot really, we cannot be confident that this is or isn't the case, right? So it could still be compatible with a salt and pepper. You could still think that there's an area that still has tuning to a particular temporal integration, but some set of neurons within that area would code for auditory, and with others, and other set of neurons would code for visual. And then the best experiment would be one where you actually, put these 2 to compete in the same experiment, which again we didn't do, right?
Yeah,
that would be the most, sensitive way to do it.
Cool, thanks.
Great, thank you, Lucia and Lisa. And